I'd like to begin by acknowledging the traditional owners and custodians of the land on which we meet today, the Wurundjeri people of the Kulin Nation. And as a research scientist, I would like to specifically acknowledge the rich culture of curiosity and knowledge building that has been active in all our indigenous communities for tens of thousands of years. It is my great pleasure to welcome today and introduce Alisa Glukhova, one of our newest faculty members in the structural biology division. Alisa did her PhD at the University of Michigan and then came to Australia to join the Monash Institute of Pharmaceutical Sciences just up the road, where she quickly distinguished herself as an outstanding GPCR structural biologist, publishing high-impact work in Nature and Cell, among others. We were delighted to recruit Alisa to WEHI in 2020, and since then she has been working to establish and grow her group in the midst of 2 mat leaves and a pandemic, as if striking out on our own scientifically isn't challenging enough. Alisa has already become a valued collaborator at WEHI, contributing to cryo-EM analysis with several studies, including with Waihong Tam and David Commander's groups. And she also chairs the Seminar Prize Committee here at WEHI, which I know is a very rewarding duty, and it means she really has to be across all of our science. Alisa received a prestigious CSL Centenary Fellowship in 2021, and she was last year's recipient of the Gottschalk Medal from the Australian Academy of Sciences, which recognizes an outstanding early career researcher for exceptional research achievements. Alisa's research program at WEHI is primarily focused on Wnt-frizzled receptor signaling in cancer. However, she will not talk about that in this talk. As you see, she's going to tell us about structure and function of 12-lipoxygenase in platelets. Alisa was kind enough to step into a canceled seminar slot and seize the opportunity to give a rare faculty talk that is not part of a review for reappointment, but just for the sheer joy of sharing a beautiful piece of recently published work, or as Alisa put it yesterday, just for funsies. So please welcome Alisa to the stage.
Thank you so much, Matt, for this amazing introduction. I feel so much better about myself now. And yes, not a reappointment talk, so please relax, don't need to judge, so just enjoy the science. All right, so as Matt mentioned, this is not, I guess, what I usually work on, so this is more of a random project that I've picked up, but it grew into what I think is a very nice story. So yes, I will tell you today about what we learned from cryo-EM of human 12-lipoxygenases, 1-lipoxygenase. All right, so this is a very collaborative project. So in my lab, Katrina used to work on it before she left, and then Vas worked on it, and then it's also collaboration with MIPS at Monash, with the TAL lab, and also with Holman lab at University of California, Santa Cruz, and also Hollenstadt lab at University of Michigan. And before I continue, I just wanted to share with you this very brief story about how this project went to be, because again, it's not what I normally work on. And this is when I met Mike Hollenstadt at a GPCR conference, and we bonded over the fact that I did my PhD at the University of Michigan, and he worked at the University of Michigan, so it was absolutely nothing about the science, other than the fact that he mentioned that he had this protein that he took to my old PhD supervisor, and he asked for him to get a crystal structure. And John rejected him because he thought it's going to be too complicated, too hard, and might require some anaerobic chambers. So he said no. And I seized the opportunity to show that I can do better than my old boss, and that's and that was the very initial thought that went into this project, and then the science came later. Anyway, So what are lipoxygenases? So they are non-heme iron-containing enzymes. And what they do is they catalyze the deoxygenation of polyunsaturated fatty acids. So think about arachidonic acid or anything with multiple double bonds in lipids. There are many lipoxygenases out there, and they're quite old enzymes, so even corals have some. But so in humans, there are 6 different ones. So in here, I'm showing you 4, so 5 LOX, 12 LOX, 15 LOX 2, 15 LOX 1. And I should also mention that nomenclature is horrendous for all these guys, because genes are named differently than the proteins, and then depending on which papers you read, it can be 15 LOX 1 or A LOX 15. It's absolutely horrendous. But the reason they're named with this, for example, 12 LOX is the position that gets oxygenated in the fatty acid. All right, so the one I will be concentrating on is this 12 LOX. And the main product of it is this 12 HPET. I'm not going to try to actually spell this out because I always fail. So the reaction that 12 LOX does, the primary reaction, is it takes arachidonic acid. It uses the oxygen from the atmosphere to create this peroxide, so 12-HPET, and then peroxidases in a cell very quickly reduce this to create this 12-HET, and this is a signaling molecule. And so what happens, should also mention that even though it is ubiquitously expressed in human body, it is a very highly expressed in human platelets. And that's why we're interested in. So Mike Hollenstadt lab is very much interested in platelet biology and trying to understand what leads to platelet activation and thrombotic VADs, and also how to prevent that from happening. And so what happens is when the calcium level rises in a cell after activation of some GPCRs, then this attracts cytoplasmic phospholipase A2 to the membrane, the arachidonic acid gets released, and then different enzymes can use that arachidonic acid. So for example, cyclooxygenase or 12 LOX, they oxidize it in different places, creating other prostaglandins, or in this case, 12-HET. And this leads to platelet activation. So despite the fact that I tell you that 12 LOX works on arachidonic acid, it can actually work on multiple other substrates, so they're all polyunsaturated fatty acids, and it creates a range of products. So platelet activation itself is a very complex event, and multiple products of 12 LOX are involved in this process. I should also say that the exact mechanism in which 12 LOX is involved in platelet activation is not completely understood. But Hollenstadt lab has shown quite clearly that inhibition of 12 LOX can inhibit platelet activation. And so that's why they were very much interested in it. So Mike also spun out a separate company called Veralox Therapeutics. And they're trying to find a way to prevent heparin-induced thrombocytopenia. So this is a condition that happens when, in some percentage of people, when they take heparin to prevent blood coagulation. So normally you would expect that if you take heparin, then it would bind to the platelet factor 4, and this would prevent platelet activation and transformation. But in some percentage of people, and I think I read it's about 6%, I'm not really sure about the numbers, what happens is they make antibodies against the heparin platelet factor 4 complex, and this in itself can actually activate the platelets, leading to activation and transformation. So it's thrombocytopenia because those platelets actually went away to form the thromb, so overall count of platelets in the blood is low. And so you can imagine this leads to thrombosis in response to the heparin administration. It's a fairly serious condition. It can lead to death. And so Mike's lab was quite interested in developing selective inhibitors for 12 LOX. And they came up with this ML355 or Veralox 1005. And this works somewhere in this step. Again, not fully understanding which of those things are getting inhibited. Well, sorry, 12-LOX is getting inhibited, but by which way it prevents the platelet activation is not completely understood. And this is currently in phase 2 clinical trials for heparin-induced thrombocytopenia. And the idea is that to get it basically through clinical trials for HIT, and then eventually maybe use it instead of heparin to prevent thromb formation even in normal patients. All right, and so Mike was interested in understanding where this ML355 is binding, because despite the fact that it's a very selective drug for 12-LOX, it is still not ideal. It's not extremely soluble, and I think IC50 is around low, maybe sub-micromolar. And so that's where we came in. So humans have 6 different lipoxygenases. They all look really, really similar. I'd say almost identical. But the structure for the human 12-LOX has not been determined yet. There was a partial structure of porcine 12-LOX, which is the closest homolog, but it lacked one of the domains. And so that's where we come in. So we wanted to determine the structure and also to understand where the ML355 is binding. I should also say that when I got my hands on the protein, the first thing I did, I set up a trace, crystallization trace. I couldn't say I did a lot. I did 4, and I was very disappointed to not have crystals, which crystallographers here know that 4 is basically nothing when you're looking for crystals. But at the same time, Dave Tal from MIPS, he had this bright idea that, look, we think that most likely 12-LOX is not monomeric. So monomeric 12-LOX would be about 75 kilodaltons, which probably is on the low range for cryo-EM, so that's why we didn't go there first. But most likely it forms oligomers, so it might be actually a very good target for cryo-electron microscopy. And that's what we started pursuing, and that's what we succeeded with eventually. So we made 12-LOX in mammalian cells. It was really, really easy. It actually, even transient expression in mammalian cells gives you a lot of protein, so it's really easy purification, his, and then size exclusion. But then what happens after that is quite interesting. So it comes up on size exclusion as multiple oligomeric peaks, and that's what we expected based on biochemistry of other lipoxygenases. So you can see here, so it's the same species. I should also say that the ratio between those 2 peaks is always a surprise. It's a really complete surprise. Sometimes it could be all dimer, sometimes it could be all tetramer. And up to this day, we still don't know what rules this equilibrium, per se. The reason we know this is tetramer and dimer is because we performed negative stain electron microscopy, and primarily we saw dimers and tetramers. This came a lot later, so after we got our very shiny new mass photometer instrument, and then we separate those individual peaks, and should also say that they are stable. So if you freeze dimer and run it, again, it still comes out as dimer peak. And so we can analyze those individually, and we see that not only dimer is dimer, but it also has a lot of other species. So tetramer, hexamer, octamer, a little bit of monomer as well. And then tetramer, again, is primarily tetrameric with some addition of other things. If we do biochemistry on those individual peaks, we don't really see much difference. So for example, this is the liposome binding. So how much of the protein sediments in a pellet or supernatant, and kind of looks very similar. Activity-wise, so enzymatic activity, so this is oxidation of arachidonic acid. Again, mostly similar. You probably, of course, can see that the separation would be incomplete. Peaks obviously overlap, so I can't claim that each one is absolutely pure dimer or pure tetramer. ML355 inhibition, again, is very similar. So the only thing for us to do was to, well, let's try solve cryo-EM structures of both, see what we can come up with. So we put the tetramer peak on the grid, and we got a structure to 1.8 angstrom, which was really, really amazing. So actually, if we do focus refinement, we get to 1.7, which is really high for cryo-EM. But if we put the dimer peak, we can actually get 4 different structures from the same grid, from the same data collection. So as mass photometry told us, it was a heterogeneous population, and indeed it was. So we were able to get monomer, dimer, tetramer, and hexamer from the same grid preparation. And so we were basically in a unique position to look at all of those species and understand what's going on. Just before I go into those species, sorry, I apologize. Just want to take a side note to just tell you a little bit about the overall architecture of the lipoxygenase. As we expected, it looked exactly like pretty much any other lipoxygenase out there. So it has this called PLAT domain, so it's a little beta barrel. It has a large catalytic domain over here, and we saw the iron quite clearly in our cryo-EM maps where it should be. And then the entrance to the active site would be somewhere here between those two colored helices, between this pink and the orange one. This alpha-2 helix will be important later on because it moves when has some conformational rearrangements in some. But again, nothing too spectacularly too different. Basically, if we overlay other lipoxygenases, again, all the PLAT domains are pretty much the same. The catalytic domain, again, pretty much the same, with the exception of the position of that alpha-2 helix and the arch helix around the entrance into the binding site. And then the catalytic residues are also all pretty much similar. So then we try to figure out, so what exactly then, why do we have so many oligomeric forms? So what could that mean? Is it just a fluke? Do they have some physiological relevance? And at this point, some of those would be highly theoretical findings, because a lot of this, for this project, we have a lot of structural data, and the biochemistry is lagging behind a bit. So for example, for the reasons for oligomeric forms, the answer is we still don't know. But one of the possibilities could be that they might have some role in membrane binding. So for example, if you look at, so this is the the surface colored by electrostatic charge. So you can see it in blue. This is the positively charged entrance to the active site. We think that it's most likely interacts with the membrane to get that arachidonic acid out of the membrane. Then if we would have a dimer, we would probably still have it accessible. It will be on the same plane. And then if we get to the tetramer or the hexamer, those would be occluded. So it is possible that tetramer and hexamer are the storage pools of the enzymes. But again, we don't know. And also what we don't know is, of course, this is recombinant protein expressed in hex cells. So the big question is, how does this look in platelets? What are those oligomeric forms in platelets, and are there indeed some storage pools or some active pools? The other thing, though, that if you look at those oligomeric forms while focusing only on their active sites, there's actually quite a bit of variation. So this, just to orient you first, so this is the anaerobic structure of coral lipoxygenase. And because it was done in aerobic conditions, they managed to trap arachidonic acid in the active site. So this is the iron there, this is arachidonic acid. And if you look at all our structures in the same spot, we could see that a lot of those active sites are occupied by something. Obviously, at this point, I can't really tell you if they're different or the same, but this is another difference between them. And this is actually something that we can answer further with structural biology. And this is what I will tell you about next. And if you think this is going to be death by structure, it sure will be. So be prepared. All right, so I will start with the hexamer. So overall structure, we got it to 2.6 resolution map, so it actually looks a little bit like pink one if you squint a little bit. So it's basically a donut, so it's a 6 hexamer, so it's 2 donuts in D3 symmetry. So our pink one would be 6 dimers, this is basically 6 monomers. All right, so they are stabilized by a disulfide that we can actually also see quite clearly in cryo-EM maps. Again, it's a cytoplasmic enzyme, so I'm not really sure how physiological this is, but it definitely is in there. What we also can do is we can take this map and then separate individual subunits and then overlay them together and basically push the resolution further, so in this case to 2.2 angstroms. And the reason why I'm telling you this is to not flex about the resolutions that we can achieve, but it is quite important if you're looking in the active site. So we're trying to look at what is happening in the active site, and so trying to understand how the small molecules are binding, if there are any small molecules. For that, it's quite important to have as high resolution as possible. Every 0.1 angstrom would count. And I shall say that at 2.2 angstroms, our confidence would be quite high in any conclusions that we would make. All right. So because we were able to push it to such a high resolution, we can actually see the density a lot better. So the density that I showed you in the beginning was much improved. So here it's shown in gray here in that map of the monomer from that hexamer. And so if we zoom in, so there are actually two discontinuous densities. So one of them is near the catalytic iron. So this is in the same exact position that was arachidonic acid bound to the coral lipoxygenase. And then another density actually can be fitted with ML355. So this is the molecule that we were looking from the very beginning. And if you hopefully can see, at 2.2 angstroms, that fit was really, really good. So it gave us very, very high confidence that it wasn't just a random blob that we placed the molecule we wanted to place, which is usually a big problem in cryo-EM field. All right, so if you look at the actual context, they've made a lot of sense. So there are a lot of hydrophobic interactions, and then we have a few hydrogen bonds. But they all really, really made sense. I should also say that this pose of ML355 is very much unexpected. So previous docking experiments suggested that ML355 would bind in the same spot as arachidonic acid would be, so that it would be competitive inhibitor. But in our hands, it's not. I should also say that I don't think one of our collaborators, so Ted, still believes that this is it, mostly because he done the docking studies. And so we agreed that at least there might be multiple binding poses. So this is at least one of them. We don't know if this is the only one. So of course, we wanted to confirm this by mutagenesis, as everybody in structural biology would do. So the complication here is if we were to start mutating residues around here, we would most likely affect the catalytic, the catalysis of the enzyme, because that's where the very much near the entrance to the active site. All right, but nevertheless, we picked those mutations. So L589, and then those four, so those are the positively charged residues. We picked them because they were as far away from the arachidonic acid as we possibly could do. And also we mutated them either to alanines or to the residues of the 15 logs, which is very closely related, but was not, has a lot, does not bind to ML355. So out of those, unfortunately, most of those residues affected the catalytic, so the enzyme rate, so we couldn't actually assay them. However, one of them did exactly what we predicted. So it fully uncoupled the catalytic activity, I'm really blanking of enzymatic activity from ML355 inhibition. So it's as good as the wild-type enzyme in terms of the oxidation of arachidonic acid, but it cannot be inhibited by ML355. Of course, I understand that one mutant is not much. So as I mentioned, the biochemistry is lacking a little bit. So we will be going back and making more mutations, so maybe separating those quad mutations into individual amino acids to fully characterize this. But it looks very promising. And also, because we do have mass photometer, we could actually put this protein on the mass photometer. And what we found is actually we basically lost that hexamer and octameric species that were present in wild type, which, again, since we only saw ML355 binding in that hexamer species, would be quite consistent with what we observed. So basically, we have quite a lot of evidence that that is indeed ML355 binding site, but we are looking forward to characterizing it further. All right, so now I will jump onto the next allogameric species, so the tetramer. And the reason I'm doing it is because they're actually indeed all different in terms of what's happening in their active sites. So this was actually really cool. So tetramer. Not going to go through this. So this is just the standard cryo-EM data processing pipeline. The only thing I wanted to point out is that if we focus on individual subunit, we can get the resolution up to 1.72 angstroms, which again is really, really high. And I think for this specific project was absolutely necessary. So just to give you a perspective, at that kind of resolution, we started to see rings we started to see holes in the aromatic rings. So here's tryptophan, phenylalanine, and it's actually quite rare to see those kind of things in cryo-maps. So I thought I picked in my cryo data processing of that, or rather Jesse did. We actually did this, but it's really, really cool. So how did the active site look like here? So we saw this, again, this continuous density on the interface of two subunits. So this is one colored in red, one colored in blue, and then we saw this. And, you know, I've seen this on multiple structures because we saw this after we incubated it with ML355, because obviously we thought, oh, maybe that's where it would be binding. However, we saw the same thing if we not put ML355 there at all, so it clearly was just something coming from the cell. And how do you get something that just looks so weird at the interface? And also, as many crystallographers would know, things that are binding in the interface would have so many artifacts that it's almost impossible to figure out what they are. We did know that it's not ML355 because it was present in the no ML355 samples, and also because I really, really tried very hard to put ML355 in there, and it just wouldn't fit. Wouldn't fit in terms of blobs, wouldn't fit in terms of interactions, just nothing made sense. It looked, can it be two head groups? Why would it have this continuous tail? Is it symmetrical? We just had no idea. This is the density. But what we could do is something similar that we've done with the hexamer. So we can expand the more videos are coming. What we did notice, though, with the tetramer is that it was very, very flexible. And so that's the analysis that we can do with cryo-EM using 3D variability in CryoSpark. So this is all the movements that you can see. So different types of movements. Just to show you that it's very, very, very flexible. And then if we do the same analysis, looking at the active site to look at that density that I'm talking about. So it's shown here in gray between these two symmetrical subunits. So in class 2 and class 3, nothing's happening, things are just moving around. But class 1 is actually really interesting. So hopefully you saw that the density was basically jumping between one monomer to another monomer. And to us, this suggested that most likely what's happening is that it's incomplete occupancy, that most likely it's present in one and absent in another, and what we're happening in tetramer, it just averages out. And that's why we have such a weird density. And so we can actually deal with that by cryo-EM. So what we took is we took the tetramer, we expanded the symmetry, we looked at them as basically individual subunits. And then we separated them. We basically sorted them based on, is there stuff in or is there not? And everything that had the density, well, basically classified it, and then we assembled the tetramer back together. And from there, we actually were able to get this really nice density that now is present only in one of those monomers, and it's not discontinuous anymore, and it has a really, really amazing shape. And then again, this is 2 angstrom resolution. And so then what Jesse did is he just went to this lipid maps database and just visually looked through it with his eyes. And then he guessed, you know what? This is going to be an Glukhova A. And the cool thing is, it actually was. So this is the fit of what he decided by eye. So hopefully you can see that the fit is really, it's a really good fit. If you look at the context, again, they made so much sense. So those positively charged amino acids, like lysine and arginine, they were coordinating those phosphates. Then we have a whole bunch of hydrophobics that coordinated the tail, and everything just looked so perfect. And then we basically tested whether, in this case, allele A can be an inhibitor of 12-lipoxygenase, and indeed it was. So it wasn't a very high affinity, so of course you can see 32.2 for 5 micromolar, but it's not outside of the physiological range. And if you look at, I should probably also mention, so the reason we chose allele A is not because we could determine that from the map. So plus minus a couple of carbons is not good enough. Like we wouldn't be able to determine that just from cryo-EM itself, nor would we be able to determine the position of the double bond as well. But Ted done a panel of different fatty acyl-CoAs with different saturations, with different lengths, and he determined that allele A indeed was the best one. And that's what we modeled in the structure. And this is actually data from last week. It took us forever to do, but at the end we took our samples for native mass spec in the metabolomics facility at University of Melbourne, and they actually determined that indeed the thing present in our samples was allele A, so it matched standards absolutely perfectly. All right, and now the last bit, so the dimer. So this is, so this dimer came from the dimer peak. Again, it wasn't unique. It was probably at about the same level as tetramer and the hexamer. But it was quite interesting because it's the only one that we saw some conformational change. So their overall resolution was 2.5 angstrom. Hopefully you can see, so there are 2 identical monomers. So, and you can see that in this case, PLAT domain is very much disordered. It is because the dimer is very flexible, so we have a lot of movement between them. And if we do the local refinement of each one, we can actually get each of the subunits to much better quality. So this is the model. And then if you look at what's happening here, so where the contacts are, so it's between those alpha-2 helices. So those are the helices that line the entrance to the active site. So they're quite important for interaction, not only the dimer, but also the other species, so the tetramer and the hexamer, so there are a lot of hydrophobic residues. And overall, it's quite similar. What is not similar is, though, that this dimer is not symmetrical. So one of those subunits is in open and one is in the closed conformation. And so this is based on this alpha-2 movement. So for the open, obviously the active site is open. For the closed, it's not. And the difference is this swinging of the alpha-2 helix by about 32 degrees. And so what's happening is, in the open conformation, because of this alpha-2, the entrance is open, and so here, as you can see, the accessible entrance, and the entire thing is well, it's actually not empty, but there is a channel that can be filled with stuff. In the closed, the alpha-2 swings, and it closes off the entrance. So if you looked at the active site density, there is stuff bound in the open, and there is basically no stuff bound in the closed. The reason I'm calling it stuff is because we don't really know what it is. We didn't have enough particles to push the resolution past our 2.6 angstroms, and we just weren't comfortable to assign anything at that point. So I don't really know what it could be. It might be arachidonic acid, but it would be doubtful because our enzyme is fully active, so you would expect in aerobic conditions, arachidonic acid won't be stuck in there. It could have been acyl-CoA again, but we just don't know. So in the future, it might be very useful to collect a lot more data to understand what this is. But also I should mention that this heterodimer, in a sense, where one is open and one is closed, creates a lot of questions about the function of this enzyme. So in the beginning of my talk, I mentioned that cyclooxygenases, so CoCA enzymes, are quite important also for platelet activations. And the COX enzymes also function as heterodimers, where one subunit would be active and the other one was not. So it is quite possible that 12-LOX acts the same way. But again, it's something that we don't know yet. So this brings me to the conclusions. So 12-LOX exist in a mixture of oligomeric species that might bind inhibitors with different affinities. We know that ML355 preferentially binds to 12-LOX hexamer. We know Glukhova A is bound at a tetramer. We obviously don't know what exactly is happening with the monomer or a dimer in terms of the active site or inhibition, but it's something that we very much would like to look at. We also know the dynamics of alpha-2 helix is crucial for the activity. And then the future directions which me and my collaborators would love to pursue, especially if we get some funding for it, is to understand what is actually the connection between all those different oligomeric forms, catalytic cycle, and the membrane binding. We don't know how they exist in platelets. We don't know what inhibits them in platelets. We don't know if Glukhova A is something that's natural occurring. And we still don't know what influences this oligomer formation. We tried multiple things. We've tried salt concentrations. We've tried substrate. We tried inhibitors, and we couldn't really still figure it out. But it's something that we would very much like to understand, especially that it looks like the oligomers are somehow tied to the ligand binding. All right, and with that, I'd like to thank everybody who was involved. So lots of people in my lab, so Katrina and Vas, and at VHI in general, so Mike, who had to listen to me talking about this on all our lab meetings, Lillian and Andrew, so VHI Proteomics, so Metabolomics at UNI Melbourne, and also all the collaborators, so MIPS, TAL Lab in University of California, Santa Cruz, so Holman Lab, University of Michigan, Hollinsted Lab, and then of course Ramosiotis Center, because all the imaging was done by Harry, and we couldn't have done it without him. And then of course all the funders for this project. And thank you so much for listening.
All right. Yeah, thank you very much, Alisa. I think that was a really beautiful demonstration of the power of cryo-EM to resolve heterogeneity in samples and give you very good specific information about all your species. Plenty of time for questions and discussions. We'll start right down here.
Hey, Alisa, really cool work. Just wondering about that cysteine that was linking the molecules together. Have you mutated that cysteine that you showed? I think it was in between the stack of the hexamer or the tetramer.
That's a great question, and not yet, no.
Okay.
Definitely something we wanted to see. We also want to see if basically oxidation when converted more into hexamer.
Yeah.
And of course, we actually need to do it because so far, hexamer is the only one that showed us that it can bind the inhibitor. Yeah. And if we want to continue with more inhibitor work and they have next generation inhibitors, we can't rely on 10% of the particles being the hexamer.
Yeah.
So definitely need to figure this out. Yeah.
Yeah, and peroxide treatment of the platelets might be handy as well to see to study the oligomerization in the cells.
Yeah, yeah, absolutely.
You found this strange binding of the inhibitor in your structure, and you were very diplomatic about how you discussed this with the modeling by your collaborator. How do you think the binding of that inhibitor, as you see it in the structure, inhibits the enzyme? Does it block accessibility, or does it kind of fix a certain conformation of the protein that is not active?
Yeah, thanks, Bernard. That's a great question, too. So in So in our structure, we saw an inhibitor bound at the same time as arachidonic acid. So we were thinking that it basically acts as a plug in a sense. So the arachidonic acid binds and stays there, and then the entrance is basically blocked by ML355. So the old arachidonic acid presumably can't exit, the new one can't enter. And I can't say that it's beyond the enzymatic, beyond what the enzymology was telling us about it. So it wasn't supposed to be competitive inhibitor. It was supposed to be mixed inhibition, which is my understanding that that mode of binding is consistent with it. Again, but it's what Ted now thinks that it might be, maybe this is an allosteric binding site, and the original one is still, can be there. But when they did this docking, they used homology modeling. And if we try to do this with the structure, it doesn't fit well. It's just, the active site appears to be too tight. So now there's more conformational changes happening, but so far, with everything we've seen, it looks like a rock. So I don't know.
Do you see interactions between the inhibitor and the substrate?
Sorry, what?
Do you see interactions between the inhibitor and the substrate in your structure or not?
No. They're close, but not enough to.
I'll just follow on that with a related question from Rich Birkinshaw, which was, is the modeling being repeated in the same docking simulation, docking position that you saw? So I believe you just said that that was homology modeling and the alternative doesn't fit in your structure. So has it been tried the other way?
Thanks, Rich. And the answer is no, it has not, because it hasn't been done by us or by him directly. It was done through a third-party collaboration. So we haven't fully revisited that yet. This is more me looking at some residues that appear to be in a different conformation, that no way they would fit. But yeah, I can't fully exclude that possibility.
That CoA that you picked up, does that make sense in terms of metabolic signaling that you know, like the role that it's playing or the synthesis of it or something? Because it seems very specifically however many carbons that is. And is that a mimetic of any kind of your inhibitor that you're using? Or is the inhibitor a mimetic of that?
Thanks, Toby. So the inhibitor looks completely different. So it doesn't have that fatty acid tail at all. In terms of physiological relevance of acetyl-CoA, to be honest, we're, again, the same answer to everybody else. We have no idea. It doesn't fully make sense because 12-LOX, it didn't look like there would be some offshoot signaling from the synthesis of acetyl-CoA. And of course, we would love to follow up, but I think the first thing to do was to see whether it's physiological in a sense. Does it happen in platelet? Like if we pull 12-LOX from platelets, is it still bound? And then, yeah, we would love to figure it out, yeah.
It's a related question. How are you planning to look at what the physiological oligomeric state is? And these are really cool structures. Is it helping you with setting that up, the information that you're getting?
Thanks, Maria. So in terms of physiological, one of the aims of my grants that I proposed is we would basically get the antibodies for the 12-LOX, and we will pull it from the platelets directly, and then use them for mass spec to see that now that we can identify, possibly get structures, because I would love to, mass photometry structures, all that kind of jazz. Yeah. Did that answer your question?
We have several questions online related to the oligomeric forms as well, so maybe I'll sort of group them together and say, have you tried to shift the oligomeric state? It looked to me like the tetramer at least was pretty stable because you dilute it all the way down for mass photometry and it's still tetramer. There's questions about, do any of the substrates or inhibitors push the oligomeric state around?
So they appear to be stable in the sense that if you collect each individual peak, freeze it, thaw it, throw it in a grid, or even throw it and put it on mass photometry or size exclusion, they all stay as they are. Different salt concentrations don't appear to change the oligomeric formation. Inhibitors and substrates, I've only done limited amount of work using size exclusion, and it didn't appear to be so, but I wouldn't be so confident. There's also a lot of issues with size exclusion for looking at it, like dilution and the time frame, but something that we definitely need to figure out.
I'll just add one of my own before I go to Peter over here. So if one thing that I don't think you addressed directly is, does that L589A mutant, does the effect of that mutation on binding actually distinguish between the two binding orientations of the drug? Does it make sense with both of them?
That's a good question. I'm trying to think. We picked it because it's supposed to. Yeah, I think the answer to your question would be yes, because we picked it far away from arachidonic acid binding, and so we would expect that it would. Basically, the competitive binding, I don't think we could actually test in our assay, because if it's competitive and we abolish that binding, we would abolish all the enzymatic activity. So the fact that we were able to uncouple the enzymatic activity from inhibition suggests that we're right. But I'm just really cautious, because it's one mutant, and I'm just cautious about making any sort of conclusion from a single mutation that worked. So I think we definitely need to do quite a bit more work in there.
Back to the oligomer. So in your gel filtration, you also had quite a big void peak. Did you ever look at that to see if there were even higher order oligomers that might be forming?
That's a great question, and no, we have not, and we definitely should.
Great. Thanks so much for that beautiful work. In terms of the physiological relevance, which is always pretty tricky to make out, do you think localization also plays a role in the oligomeric form? Because what you perhaps could look at is a cryo-claim workflow, which is extremely tedious, but if you somehow could localize them, mother down with like cryofib, and then focus on that section. Is that something you're looking at or to consider?
Just described a proposal.
Oh, wonderful. Okay.
But yeah, definitely, because if we're proposing that some oligomers can't bind membrane in their storage pools, then the logical extension that the localization should change between membrane and cytoplasm. So definitely needs to be looked at.
All right. If there are no other questions in the room, I think then we'll bring it to a close, and it just leaves it to us to thank Alisa again for a great talk and for stepping in at relatively short notice.